afternoon, everyone. My name is Samantha Case. I'm a PhD student at NC State in the Department of Plant and Microbial Biology. My PI is Dr. Zhu Li, and my interns and I would like to present to you about the research we've done over the summer about the association mapping of antioxidant capacity, consumer aesthetics, and taste in blueberry. But first, I would like to introduce my awesome interns. We have Estefania, Ashley, Jordan, and Brian, and they have been instrumental in helping me advance my thesis. A brief presentation outline. I'll go over some of the background and human health impacts of the two crops I study. I'd like to go over some of the research purpose of my thesis. I'd like to summarize my broccoli GWAS results. And then my interns will go into the background of the antioxidants, background of the metabolomics data we've developed, go over the results of their blueberry QTL analysis, and then we will go over the research application of all of this work. So the first crop I'm working in is broccoli, Brassica oleracea variety italica, and it has a huge human health impact due to the compound glucosinolates found in broccoli, the Brassicaceae family, and when that's exposed to myrosinase, it converts into isothiocyanates, which have been shown to be very potent anti-carcinogenic compounds. And then the other compound that's really important in broccoli is lutein, which is really important for eye health. So the second crop I'm looking into is blueberry, which has gotten a lot of press recently for its antioxidant capacity, which has mostly been attributed to the anthocyanins, which is the blue pigment. And then it also has a lot of dietary fiber, which is really important for gastrointestinal health. Historically, crops have been bred for higher yield and longer shelf life. Like my colleague Neha has described earlier, that usually leads to a decrease in metabolite diversity. And with that decrease in metabolite diversity, we usually have increased susceptibility to pathogens and antibiotic stress, poor taste, and decrease in nutritional quality. So the dilemma is, how do we marry all these traits together and breed for those really ideal crops where we have high yield, pathogen resistance, good taste, and improved nutritional qualities? The purpose of my thesis hopes to generate data so we can start addressing this problem through genetics, and I'm hoping to do that through association mapping. So association mapping is the statistical association between genetic regions in the genome and a phenotype. A genome-wide association study uses a diverse population of unrelated individuals, whereas traditional QTL mapping uses a family-based structured population. So for my first specific aim, I'm hoping to find, using association mapping, candidate genes that regulate agronomic and hedonic, or aesthetic and taste phenotypes in broccoli and blueberry. My second specific aim is to develop a draft metabolome utilizing multiple different profiling techniques in blueberry, and then to use that draft metabolome to then find regulatory candidate genes for specific metabolites in blueberry. This summer, my interns have been instrumental in helping me advance and almost complete my specific aim 2 and 3, so it has been unbelievably helpful. To summarize my broccoli results from the past few summers, this is a traditional Manhattan plot used to depict broccoli GWAS results. And so along the x-axis is the different chromosomes, and each dot represents a genetic marker. So any dot above that red dotted line is a significant marker, thus a significant QTL. So here we have two significant QTLs for the days to harvest phenotype, meaning that if broccoli breeders were then to breed for these two QTLs, they might be able to shorten the broccoli harvest season, therefore they could maybe fit in three harvest seasons instead of just 2 in the overall growing season, which would be really good for increasing yield. This is a depiction of compiled QTLs for multiple different pathogen-related traits in broccoli. The black dashed lines are the genetic map the first-year interns at P2EP helped me create. And then each colored marker is a different pathogen trait. So we're looking for overlapping colored markers signifying the different traits, because then that indicates pathogen-resistant hotspots. So if I can focus your attention to those 4, where there's multiple pathogen traits overlapping, that means that if broccoli breeders were then to select for those 4 QTLs for their broccoli crop, that broccoli crop is going to have generalized pathogen resistance for a multitude of different pathogens, which is going to obviously improve harvest and yield. I'm going to hand it over to Stefania to describe what they've been doing over the summer.
Thank you, Samantha. So before we get started, we want to give you a brief background on what an antioxidant is and what it does in order to better illustrate why this research is so important. So in a healthy human body, our atoms come with paired electrons, which means that each electron has a partner electron, and this is how our atoms are most happy and most healthy. But sometimes something called a free radical comes along, and what this free radical does is that it attempts to steal an electron from a healthy atom, and it does this because it's missing one of its partner electrons. And so this is what we don't want, because this can cause a number of health concerns and issues. And that's where our antioxidant comes in. Our antioxidant is kind of like a superhero. It swoops in and it tries to donate an electron to this free radical so that it no longer feels the need to steal electrons from healthy atoms. And the reason that we're focusing on blueberries this summer is because blueberries are known to have a high number of antioxidants. And now Ashley's going to tell you a little bit more about how we've been doing this in the lab.
Okay, first and foremost, the issue that we've had to address is the fact that how do you measure antioxidant capacity, or how do you measure the superhero's powers? To do this, we optimize the DPPH protocol. DPPH is unique in that it has a stable free radical. When in the presence of an antioxidant, it will convert to its non-radical form, as indicated through a color change from a dark purple to a pale yellow. The diagram below will just show you an assay, an example of how we plated our samples. After running these samples through a spectrophotometer, we achieved the following results, which is displayed graphically behind me. The solid black line, that is our Trolloc standard curve that we established, and the purple dots represent the 214 blueberry samples that we tested. I'd like to draw your attention to the bottom half of the graph, in which there are 3 dots that stand out the most, and those represent samples that were high in antioxidant capacity. And now I'll pass this on to you.
Thank you, Ashley. The LCMS shows secondary metabolites, specifically anthocyanins. The different colored lines represent the 9 samples. The peaks also represent the anthocyanins. The heights of these peaks show that there's variation between samples within a certain anthocyanin, and this variation is important because we can then use QTL mapping to locate the region that is associated with anthocyanins and thus antioxidant capacity. The NMR spectra shows primary metabolites, specifically sugars. The variety of sugars that are shown is from blueberries, and the sugars are labeled on the graph. You can also see that there's variation between these sugars, and we also did QTL mapping for these to see where the region is located, to see where it's associated with the sugars and thus taste. Now Brian will talk to you about QTL results.
So here we have a composite map of antioxidant capacity and anthocyanins. As Samantha mentioned earlier, the black lines are our genetic regions, while the colored markers are our traits. First, I'd like to draw your attention to linkage groups 10 and 12, where we have 2 different groups, both of them containing antioxidants and anthocyanins that are associated with each other due to the fact that their regions are overlapping with each other. Also on linkage groups 2 and 8, we have antioxidant capacity that is not associated with anthocyanins, where we believe there are other chemicals in play that may be associated with antioxidant capacity. Now I'm going to send it back to Stefania so she can talk about aesthetics, consumer aesthetics of blueberries.
So this is a QTL map of traits that are associated with consumer aesthetics. And this, of course, is important because if we want to increase the consumption of blueberries, we want to make sure that these blueberries are appealing to consumers. And so what we've done is we highlighted some of the more important results in orange boxes. And what we found was that berry firmness and the firmness rating are associated with, which of course makes sense and we expect to see. And we also saw berry firmness associating with percent juice, which again makes sense and we expect to see. But we also have an anthocyanin profile that we overlaid over this. And what we found there was that the color rating of blueberries is also associated with anthocyanin content, which makes sense because anthocyanins are pigments. And we also found the anthocyanin content was related to berry firmness and percent juice, which may indicate that anthocyanin content may have an effect on the berry firmness and the percent juice of a blueberry. And now Ashley's going to give you more on taste.
Thank you, Steffi. Well, Steffi talked about consumer aesthetics, and that is an important attribute. Taste is also something that we need to consider. Behind me is the taste QTL map, and I want to draw your attention to linkage group 9. You will see 2 orange boxes there, and these orange boxes display relationships between flavor rating and the ratio of soluble solids to titratable acids, as supported by previous research. The other orange boxes will highlight other places in which sugars and the acidity ratio contributes to taste. And now I'm going to hand this to Brian for conclusions.
So in conclusion, for broccoli GWAS, we found data linking to harvest time and pathogen resistance. For the metabolome for blueberries, we quantified over 300 metabolites. And then in blueberry QTLs, we found antioxidant capacity that's associated with anthocyanins and some unknown compounds, consumer aesthetics that are associated with anthocyanins, and firmness, as well as taste that is related to sugars and acids. I'll finally send this back to Samantha so she can talk about future work.
So thank you, Brian. So our overarching goal is to increase consumption and improve nutrition. Because if breeders are then to go into breed for the results that we have and some of these conclusions, they'll be able to have crops that have, more pleasing to look at broccoli, more pleasing to look at blueberries, and improve nutrition. And if these breeders can produce all of these traits that we're looking at, then consumers will consume more of these fruits and vegetables, and then they will benefit from all of the nutritional qualities that these broccoli and blueberry. And so everybody wins. Come ask us questions if you guys have any. The aesthetics and taste research will be at poster number 1, and the antioxidant capacity will be at poster number 8. Thank you for your attention.
Thanks everyone for your attention. Right now we're going to have a short break. There's some refreshments out in the where the food was. Just on the other side of this wall will be the poster sessions. So feel free to walk on over, check out the posters, see the students will be presenting their work in a formalized scientific manner. Anybody who's been tagged as a judge, if you can meet me over at this corner, and I'll explain the judging rules to you. We will be meeting back here at 2 p.m. All right, I think that's everything. So.